Okay, welcome everybody. My name is Sophie Thomas and it's my pleasure to welcome you to our third Fabry webinar focused on gene therapy. This is a very interesting talk. Obviously, gene therapy has been spoken about for many years, about 15 to 20 years, even before enzyme replacement therapies came on board. And we're now at that stage where we're having actual gene therapy trials, where we're in the process of recruiting patients to these trials. So it's a very exciting time, and I think that the aim of this webinar is to really inform you about the different types of gene therapy and what's involved. And we're very fortunate to have a patient speaker as well joining us. Okay, next slide. So before we move on, just some basic housekeeping. We do ask you that you do not take screenshots. We also ask that you don't reproduce any slides without the speaker's consent. There will be a question and answer session at the end. We do have a question and answer function button on the webinar, so if there are any questions as we go through the presentations, then please do send them to us. We will try to answer as many as we can, but we may not be able to answer them all. The webinar will be recorded and available via the MPS Society shortly after this webinar. Thank you. Next slide. Okay, so it's my pleasure to introduce you to a number of speakers. Online, we do have Professor Atul Mehta, but unfortunately, due to some technical issues, he may not be able to join us live, but he is here listening. And if there's any questions specifically that he can answer, we can direct them to him. We also have Professor Brian Winchester, who's going to give us an overview of gene therapies and their deliveries. We've got Professor Darylyn Hughes from the Royal Free Hospital, who's going to give us an overview of the in vivo gene therapy. We also have Professor Michael West, who has joined us all the way from Canada, who's going to talk about the lentiviral gene therapy. And as I mentioned before, we've got Ryan DeVoe, who is a patient who has actually received gene therapy through one of the clinical trials. Okay, so next slide, please. And the next slide, please. So it's my pleasure to hand over the reins to Professor Brian Winchester, who's going to give the overview of gene therapies and how they are delivered. Thank you, Brian.
I'm on my phone, Sophie, so I'm not quite sure if you can hear me. Can you hear me?
Yes, we can hear you.
Well, lovely. Well, maybe I'll just say thank you very much, Sophie. And it is a fantastic program you've set up, because you've got Brian, and we all know Brian, who talks straight and talks to patients. And then we've got Darylyn, and that's fantastic because she's got first-hand experience of gene therapy. And we've got internationally, we've got a world authority in Mike West on Fabry disease, and that's fantastic. And then, of course, you've got somebody who knows more about it than the rest of us so-called experts put together, and that's a patient who's actually had gene therapy. So it's a thank you to yourself, Sophie, and the team for setting this up. And congratulations to the Fabry community, including, of course, the patients, for getting us this far. You know, 21 years ago, we had nothing specific for Fabry. And then we've come up with enzyme and chaperone. And who would have predicted gene therapy? Well, exciting. So I think that's over to you, Brian.
Thank you. Have the first slide then, please. I think it's important for understanding of gene therapy for Fabry disease that we look first of all at the genetic basis of Fabry disease. And on the left-hand side, you can see the normal situation. Here's a patient who is unaffected and has a gene called the alpha galactosidase A gene, or GLA for short. And this provides our cells with the information for making that enzyme, alpha galactosidase A. Now, alpha galactosidase A plays a very important part in our metabolism. It breaks down and recycles sugary fats called glycolipids, and it prevents them accumulating inside our body. And that is the situation that happens in Fabry disease when that enzyme is missing, that these sugar molecules accumulate inside the body. And if we move to the right-hand side, you can see there's a Fabry patient, and the gene, the GLA gene, is altered in that patient. It's mutated. And as a result of this mutation, no enzyme is produced, or very little enzyme is produced, or in certain circumstances, you get a faulty enzyme that doesn't work properly. Whatever the cause, there's not enough enzyme activity, and the glycolipids accumulate in our cells and tissues. And you can see the picture of a kidney cell with globules of fat actually inside the kidney. Or you can see an angiokeratoma, where accumulation of these fatty sugars has caused the blood to spill out and form a keratoma. So Fabry disease is a result of a mutation in a gene leading to the absence of an enzyme, leading to the accumulation of these sugary fats, which then lead to a whole series of other events. So what can we do about this? Next slide, please. What forms of therapy are there for Fabry disease? Well, one thing we could do is replace the missing enzyme, actually inject the enzyme into the body of a patient, so-called enzyme replacement therapy. Another way of introducing the normal enzyme into the patient is to transplant cells that produce the normal enzyme, a bone marrow transplantation from a donor. Those cells from the donor make the enzyme and give it to the patient. For those patients who have a faulty enzyme, we may be able to add a little molecule, a drug that causes it to fold up correctly and have activity and restore the activity in that way. And that is chaperone therapy. We might be able to also just decrease the amount of those glycolipids in the patient so that it doesn't build up so quickly. And this is so-called substrate reduction therapy. But this evening, we are talking about gene therapy. And that is the addition of a copy of the normal gene to the patient's cells. Gene therapy. Next slide, please. So gene therapy is the addition of a copy of a normal gene to the patient's cells. Now we can make the normal galactosidase gene in the laboratory by genetic engineering. But if we just injected that into the body, it would be broken down and would never reach our cells. So we have to put it in a carrier, a protective carrier, before we put it into the patient. This is a carrier or a vector, and is usually a virus. Although it's very interesting, from the COVID epidemic, we now know that we can use other forms of molecules to introduce genes into patients. And I think we will see that developing also for metabolic diseases as well. Okay, so we put the normal copy of the gene into the virus. The virus is modified to make it safe. We don't want to give a person a disease due to the virus. The virus is crippled, so it cannot replicate, but it can still infect human cells. It can deliver the good, normal human gene to the patient's cells. And then the gene is expressed and the patient's cells make the enzyme, they correct themselves, but they also secrete the missing enzyme into the patient. So gene therapy, as it's practiced now, takes two forms. Firstly, in vivo gene therapy, that is direct injection of the viral vector into the body. And the second form is what's called ex vivo gene therapy. We take cells out from the patient, add the corrective gene to them outside the body, and then retransplant the corrected cells back into the body. So two forms of gene therapy, in vivo gene therapy and ex vivo gene therapy. Could I have the next slide, please? First of all, in vivo gene therapy, and we're going to hear about this a lot more in practice from Derelyn Hughes. So the gene is put into the virus, and it's usually an adeno-associated virus, which has been modified to make it safe. It's injected into the patient. The virus particles enter the human patient's cells, and then the corrective gene is switched on. It corrects the cells that have been infected, but also, as you can see, it secretes enzyme, normal, good enzyme, to go around the body. And this enzyme can be taken up by other cells and tissues in the body. So the targeted cells are corrected. They then make enzyme can be exported and taken up by other cells by the process of what's called cross-correction. Next slide, please. But we can also direct the vector to specific targets in two different ways. Firstly, by the route of administration. We could inject the virus containing the gene directly into the brain, and this is being tried out. Or we could direct it into another organ. Say we wanted to correct the metabolism in the muscle. We could direct the gene directly into the muscle by injecting into the muscle. Next slide, please. We can also target the virus to particular cells or tissues by choosing a particular strain or variety of the virus. This virus, adeno-associated virus, exists in many different forms, and they naturally move towards certain types of tissues. And we can also modify the surface of the virus so it goes to a particular type of organ or tissue. For example, we could use a strain or a serotype, as it's called, that is targeted towards the heart. And I tried to show here that we could deliver the virus and the gene directly to the heart. There are strains or viruses that will actually cross the blood-brain barrier, and we may be able to direct the virus through the blood, through the across the blood-brain barrier to the brain. And all these things are being tried out now, both in clinical trials and in experiments in animals. And we're going to hear a lot more about that from Derelyn later on. Next slide, please. What about ex vivo gene therapy? This is the transplantation of blood stem cells containing the normal gene. Now, in normal bone marrow transplantation, the cells are obtained from another person who is making the enzyme, and then they're put back into the patient. But with this approach, you take the patient's own cells out. Say bone marrow is removed from the patient, it's grown in culture, and then it's infected with a virus containing the normal gene. So the patient's cultured cells are infected with the vector and start making the protein that's required. After purification and checking the cells, the corrected cells expressing the normal human gene are returned to the body by transplantation. And these modified cells then start producing the enzyme, which can be distributed throughout the body again by secretion and uptake. Next slide, please. Now, the cells that are used in this process are called stem cells. Now, these are cells which have the ability to change into other cells. They are precursors of different types of cells, and they're extremely important in the body. We have about 200 different types of cells in the body, and they're all derived from one unique cell. And gradually, as they go along, at different stages, there are stem cells that give rise to lineages of other cells. So these stem cells that are transplanted can change. They can differentiate into other types of cells and migrate to other parts of the body. And I've tried to show in the figure here that they differentiate into different types of cells that move to different types of the body. And one type can actually, again, cross the blood-brain barrier. And so we have a means, perhaps, of delivering enzyme and gene to many different parts of the body by one transplantation. Now, the second very important point about ex vivo gene therapy is that we are actually using the patient's own cells. So this decreases considerably the immunological problems of a transplantation. It doesn't eliminate the problem is the protein which is not made by the patient immunogenic, and that's something that all therapeutic strategies have to take notice of. Now, there is one very exciting development that's not actually in clinical practice yet, but is coming on. We can now make stem cells from ordinary mature cells like skin cells. We can make them go back to stem cells, and we can probably make stem cells that will differentiate into every single type of cell in the body. And this process is called induced pluripotent stem cell production. And it's something which is going to be very important in the future, because we can take the patient's own cells, differentiate them into these very many different types of cells, which could then go back into the body and change into almost any type of cell within the body. And I think this is going to be a very exciting future development. So ex vivo gene therapy, in summary, is really bone marrow transplantation of the patient's own cells that have been corrected by gene therapy outside the body. And we're very lucky. We're going to hear from Michael West later, and from Ryan, who's actually received some of this therapy. And so I'd like to finish at this point to say that we are at a very exciting stage in gene therapy. There are examples of in vivo and ex vivo gene therapy, and on the horizon are some very sophisticated new techniques using not viruses, but lipid particles to deliver genes. And we have the potential of almost an infinite number of stem cells that could correct different parts of the body. And at that point, I'd like to finish and hand over to Derelyn, who is going to talk about in vivo gene therapy.
Thanks, Brian. So it's great to be with you all this evening to talk about gene therapy. And I'm going to be talking about in vivo gene therapy. I'm going to start with a brief overview of in vivo gene therapy, and then I'm going to tell you about some of the specific studies which are ongoing in the UK currently. So these are my disclosures. The next slide. Thanks. So the reason why we are really very interested in developing new therapies for Fabry disease is to make people with Fabry's lives better and longer and easier. And there are a number of things that we need to do to achieve that. So enzyme therapy and chaperone therapy, which are available now, have had really great effects for patients. But we know that there are some things that we could improve on. So some people who receive enzyme may get antibodies, and that may cause reactions, or it may reduce the effect of the enzyme. We know that regardless of how long some people have enzyme, there may also be problems in the long term with some of the function in the organs still progressing. We know that enzymes don't address all the mechanisms of disease, and they don't necessarily get into all of the tissues. And of course, we do want to make the treatment more convenient for people. Next slide. So gene therapy is just one of a number of ways that we're currently exploring to meet those aims of making people's lives better and longer and easier. And Brian has outlined beautifully what gene therapy actually is. So replacing a mutated gene that causes disease with a healthy copy of the gene. There's some other forms of gene therapy which are useful in other conditions where a gene that is doing something bad in itself can be knocked out, or we could introduce a completely new gene in the body to help to fight a disease. Next slide. But for Fabry disease, we're talking about replacing the missing or defective gene. Now, you can see there a little diagram of some DNA, and you can see the bit that's wrong, the bit in X's. So the curly bit is the DNA, and the X's are where the DNA has gone wrong. And this is where the mutation is in the Fabry alpha-galactosidase A gene. So what we need to do is to replace that. Now, at the moment, with the technology that we currently have, we're not going to remove the bad gene. That might be something that in the future we feel that we need to do, but we're going to give a copy of the good gene so that we can make enzyme to treat people. Next slide. So I'm just going to recap some of the things that Brian mentioned so that you understand the context of what we're trying to do with in vivo gene therapy and what the current studies are all about. So you'll recall that with in vivo, which means in the body gene therapy, we're going to give the DNA wrapped up in a virus directly into the patient. Next slide. And the way we're going to do that is by using a little virus. In this case, the viruses are called adenovirus or adeno-associated virus as a spaceship. And in fact, you can see the viruses do look a little bit like spaceships. So we're going to put the DNA inside the spaceship and inject it into the body. Next slide. So the virus is made in living cells in a laboratory. So the cells grow the virus and the DNA is put into them. Then there's lots of quality control checks, and the virus with the right gene inside is harvested. So it's taken out and put into a bag, just like your enzyme is in a bag, just the same. Next slide. And then the patient who comes up to our clinic for the clinical study has an infusion of the virus, which is containing the DNA for the enzyme. And it happens once, it's a one-off infusion. And of course, that's what's going to make this a very much more convenient treatment in the long term. So once gene therapy is working and approved and we've got all the things right about it, it will be a one-off treatment that will last, hopefully, a good number of years. It may last indefinitely, but we don't know that yet. But the intention is it will last a long time. So the infusion will go into the body, into an organ. The virus will grow there, and the DNA will start to make the enzyme. And in the end, the virus itself will go. The virus will die, but the enzyme itself and the DNA will continue to be there in the patient's cells and make the enzyme for the long term. At the moment, the in vivo gene therapies that we have are directed against the liver. That means that the virus goes to the liver and then the enzyme is made from the liver and goes into the bloodstream and moves around to various important organs, such as the heart, but other organs in the body. In time, it might be possible to have viruses which go to specific organs and genes which work in specific organs. So the enzyme is made within the organ rather than made in the circulation. But at the moment, the two studies that I'm going to mention, the viruses both go to the liver. Next slide. Okay, so here it is. The virus has got the enzyme DNA in it. It goes to the liver and it makes enzyme which goes around the body. The important thing is that the bad bits of the virus that make it a virus, that make it give people whatever the symptoms of the virus are all stripped out. So the virus is no longer active except as a little spaceship to carry the DNA around the body. Next slide. Oh, there's my DNA. Next slide. Okay. So although these viral vectors, these viral spaceships are created from viruses which can in normal circumstances cause human disease, they're actually modified in a way that makes the minimum harm. So they're modified to make them safe. Sometimes, because it's still got a bit of virus protein there, people get some inflammation and there can be some side effects. But the idea is that those will be minimum from the virus point of view. Next slide. Okay. So viral in vivo gene therapy has been used in some conditions now for a good number of years. And I show a picture here of one of my colleagues, Amit Nuthwani. So he works in our department at the Royal Free. And he spent his life building gene therapy for bleeding disorders, people with haemophilia. Haemophilia is also an enzyme deficiency, but here the enzyme would normally cause blood clotting. If you don't have it, it causes lots of bleeding. He's worked on gene therapy, and some patients now have had in vivo gene therapy using this adeno-associated virus that goes to the liver. It's producing the normal factor that helps blood clotting over 6, 8 or more years. So that's shown us that there's a lot of potential with this sort of gene therapy. Next slide. Okay. There are some risks, of course, as with anything. So there's some risks of developing antibodies to the virus itself. Now, if that happens, it might cause there to be an immune reaction transiently in the liver while the virus is still there. And that might be something that we need to treat as part of the gene therapy protocol. We can develop antibodies to the virus. We might make antibodies to the enzyme as well. Now, that's not something in gene therapy that's been documented, but it is a theoretical possibility. We might get inflammation to the liver cells, and we could get virus in other cells. There's also a possibility that the DNA, depending on where it goes within the cell, might cause cancers in the future. Again, these are all theoretical, and everything is designed to try to prevent this from happening. And there's also the risks, of course, of having blood tests, which you would get with any sort of study. Next slide. So the potential benefits for gene therapy are that by administering the gene therapy, we manage to elevate the level of enzyme for a longer period of time. So on this graph, you can see that we've got little peaks of enzyme every 2 weeks. The idea with gene therapy is with a one-off infusion, we manage to elevate that level. So if you just click again, so that's what we might be seeing with a continuous level, for example, of chaperone therapy. And then click again. That's what we might see with gene therapy, but that line could be anywhere on that graph. We don't yet know where that line will be. It could be very high or it could be lower, depending on how well it works. Next slide. Okay. So those are the principles of in vivo gene therapy. We give a virus which contains the DNA and is stripped of all the bad bits. The virus goes to the liver, makes the DNA makes the enzyme. Eventually the virus itself goes away and the enzyme circulates around the body and treats the patient. So it's creating its own factory of enzyme therapy within the patient. So in the UK, there are currently 2 studies that are open and recruiting for in vivo gene therapy. I'm just going to briefly tell you a little bit about this, and all of this information is from the clinicaltrials.gov website, and you can find information on any study in Fabry disease on this website. So the first one is the Freeline study. It's called Marvel 1. And the vector here is called FLIP190. doesn't matter what it's called. And this is an adeno-associated virus. And the bit that's in inverted commas there is what Freeline say about this vector. So that's on their website. You can also find information from the company websites. So patients come up to the site and have quite a lot of investigations. There's various radiology and biopsies and blood tests and so on. And then they have the infusion of the virus. And then after that, there's quite a lot of follow-ups to see how well it's working, measuring the enzyme activity and various other parameters. There's two cohorts that will be, two groups of patients that will be recruited. The first group are people who've been treated before with enzyme. And the second group will be patients who've never had any treatment. Next slide. The main inclusion criteria for this study are that the patients are male, that they're adults, and that they have classical Fabry disease. And by that, we mean that they've got multiple organ involvement with usually pain, rash, sweating problems. So this particular study at this time is not recruiting patients who have late onset Fabry, which is mainly cardiac problems later in life. Next slide. There are some other inclusion criteria. I've put this up because this can go on the website. I'm not going to go through it all now, but there's some other things that we have to tick, mainly to make sure that people are affected enough to potentially, if the treatment works, to see a measurable difference. Next slide. The exclusion criteria, this means reasons why people couldn't go in it, is if they have non-classical Fabry disease, if they've got antibodies to one of the current treatments, in fact, also if they've got antibodies to his adeno-associated virus, because that would mean the body would just reject the virus. And then if they've got really bad kidney or heart disease, or they've been on another study. Next slide. Again, there's lots of other exclusion criteria. I'm not going to go through all of this, but it's a little bit of, dotting the I's, crossing the T's, and make sure that people are well enough to go on a study. Okay, next slide. So the second in vivo study, which is open in the UK, is a Sangamo study. And this is actually sort of seems quite similar when you read it. Again, this is from clinicaltrials.gov. It's an adeno-associated V2/6 virus. And again, the idea is to provide long-term production of alpha-galactosidase A for patients with Fabry disease. And again, patients are infused on day 1 and then have follow-up for a year. Both of these studies have a lot longer term follow-up, so we can understand what happens with gene therapy in the long term. Next slide. Very similar inclusion criteria. Adults with Fabry disease with one or more of the following symptoms, similarly sort of a classical Fabry disease. And the exclusion criteria on the next slide. Again, antibodies or being on chaperone therapy or having bad heart or kidney disease. Next slide. And then there's a range of other exclusion criteria, which are sort of, some are similar, some are different. Next slide. Okay, so those are the two studies which are open in the UK. The way to get more information about those, if you don't want to look on the website, so to talk to your physicians, the adult physicians at all the centres know about these studies and will be happy to talk to you. And if it's not yet open at their sites, they'd be happy to refer you. There's a plan for all the sites to be open for one or more of the studies, but sometimes it takes a while to open gene therapy studies because of the regulations. So I've just got a couple of summary slides. So possibilities for gene therapy, why might this be helpful? Well, it's possible in the future that enzyme might get into the brain, might require specific vectors to do that. It might be quite convenient to have a one-off infusion, although you should remember that if you're on a trial, there's lots of coming backwards and forwards, so lots of tests and investigations during at least the first year. There's obviously some risks associated with both. Probably less of a possibility for antibody treatment with gene therapy, maybe more of a potential for getting it into tissues. And then the lifetime costs, we don't really know. Next slide. Next slide. Okay, so there's some challenges for us. So things that we need to understand. So how is it important that the enzyme stays in the circulation, or that it's made in the individual tissues? How does it get into the tissues? How can we predict how well it's working? And then at the end of the day, how reversible are some of the changes that we see in Fabry? We know that early treatment is beneficial for people, and that is by far what will make the biggest difference overall. Next slide. So these are some questions for me as I think about these studies. So what sort of gene therapy should we be thinking about for our patients? And is it different for different patients? Should we be using in vivo gene therapy for some people and ex vivo that Professor West will talk about? Which virus? Is there a difference? We need to understand that. Which organs? What is the dose? And all of the studies using gene therapy start off with low doses and then gradually increase the dose to get to the right dose. So that's something we need to understand. When you have the gene therapy, most of the studies require some degree of immunosuppression round about the time when you have the infusion. And that's to prevent you getting inflammation in the liver. So that's also an important thing to think about. And then the final important thing, which we really don't know, is what happens in 10 years' time. Will there still be enzyme there? And if there's not, can you have another treatment, the same one again or a different one? We don't know yet. This is the long term. The next slide, this is the final one. So we talked about gene therapy being on the horizon for many years. And I think the horizon is now very much more with us. In fact, we have patients, a patient kindly here with us tonight who've been treated with gene therapy. So this is a reality for us in clinical trials to think about. And I think that's a great thing. It's an amazing thing, as Atul said, over the last 20 years, we've come a long way. And this is the start of a new journey, which will hopefully make lives better and longer and easier for patients with Fabry disease. So thanks, Sophie, for asking me, and really happy to take questions at the end, or you to email me if you have any questions. Oh, and I'd now like to introduce my colleague from Canada, Professor Michael West from Dalhousie University, who's going to talk about ex vivo gene therapy and his experience with lentiviral gene therapy.
Thank you very much, Darylyn. It's a pleasure to be here, and I thank the organizers for the very kind invitation for me to talk about our lentiviral gene therapy study, which is well ahead of everyone else, because I think we started about 10 years ago. Next slide, please. And our study is also listed on clinicaltrials.gov, and we call it the FACTS Project, Fabry Disease, Clinical Research and Therapeutics. Next, please. These are my disclosures. I've received research funding and consultant fees from a number of the companies that are active in Fabry disease. And I also have some intellectual property in terms of gene therapy and cardiac biomarkers. Next, please. So this is our paper, which was just published in February, and it is the first trial in patients with Fabry disease with gene therapy in the world. It's a phase 1 trial, meaning that its focus is on feasibility and safety of this whole procedure rather than on outcomes. So there's still lots to be learned. And as you can see from the long list of names here, I didn't do this myself, so I won't take any credit for that. And Jeff Medine is the principal investigator and leader of this research group. So we used autologous stem cells, and you've heard Dr. Winchester talk about these stem cells, and the ones we used were stem cells from the blood. So they make blood cells, they normally live in the bone marrow, and this is an ex vivo procedure whereby the patient's stem cells from the blood are removed. They normally live in the bone marrow. We are able to trick them into moving from the bone marrow into the peripheral blood where they can be easily accessed through an intravenous needle. We can take them out and then use them to add a gene back to the patient, in this case, the GLA gene for Fabry disease. And this is the normal process, what goes on in a stem cell transplant, which is used to treat many patients now as standard of care for a number of different blood disorders and certain types of cancer. So this is a well-known procedure. It's been well studied, and it's been standard therapy really for a number of decades. So we're using a proven method of stem cell transplantation to, in this case, put a novel gene into human cells. Next slide, please. So why lentivirus instead of adenovirus? Well, the lentivirus has been used for a number of years in gene transfer experiments. With good success, it's highly efficient at transferring genetic material, that is DNA, into the host human cell. It's got a really good safety profile, and it's also been successfully used in gene transfer studies and other lysosomal storage diseases, and in particular in adrenoleukodystrophy, where there's an ongoing study in children looking at gene therapy there. The other potential advantage of lentivirus is that most people never see a lentiviral infection in their lifetime, so they don't have any antibodies to lentiviruses to worry about. So in the process of doing this autologous stem cell transplant with the lentivirus, we end up overexpressing the GLA gene, which means that the cells that are transfected with this viral vector overexpress the gene, so they overmake the enzyme. So they're making extra enzyme, and the extra enzyme can then be exported into the circulation and then travel to other cells, and should be able to be taken up by cells through the same route that the enzyme therapy that many patients with Fabry disease take up enzyme. And we know that in animal studies that if you overexpress these genes, particularly for the alpha-gal enzyme, that in mice it doesn't cause any problem, even though they may have a 10,000-fold higher level of enzyme activity. So in mice, at least in the short term, these very high amounts of enzyme are well tolerated. Whether that's the same in humans is still to be learned, I think. And as Dr. Hughes has already nicely explained, that there's reasons for us to think that a sustained level of enzyme activity above the background may be more effective than giving a large bolus of enzyme therapy, such as we give with the enzyme infusions. Cost is still unclear. But it may be that eventually that gene therapy could be less expensive than the current enzyme replacement therapy, but we don't know. So these hematopoietic stem cells, when we use them, do have the advantage that they can deliver enzyme into the brain, whereas the intravenous enzyme therapy does not cross into the brain currently and has to be directly injected that way. And the other consideration is that these blood cells themselves may be a major source of GB3, and that's this fatty material that accumulates in Fabry disease. So in particular, if these blood cells are now restored with above normal enzyme activity, then they won't be producing GB3 that's going to clutter up the other cells in the body. Next slide, please. So this whole process with the ex vivo gene therapy rests upon this concept of metabolic cooperativity. And this has been well investigated in animal models for about 20 years. And so on the left, we have a cell that's been transduced. So it's been transformed, if you will, by the gene transfer of the enzyme, as shown in the little blue bar. It overmakes the enzyme, the little orange figures there, and the excess is excreted into the surrounding area in the body, where it's taken up by the cell on the right, representing a cell that hasn't undergone gene transfer. And they're able to be taken up through receptors on the outside of the cell. They get delivered to the lysosome, where we know they can break down accumulated GB3. That's the fatty material. Next slide, please. So this is the outline of our study. This study was funded by the Canadian Institutes of Health Research. That is a independent health research organization funded by the government of Canada and therefore the taxpayers. And so I paid for a small part of this, I'm sure. So basically, we identified patients who were suitable for the study. I'll go through the inclusion criteria a little bit later. We had to get a lot of baseline tests on them to make sure that they were stable and healthy enough to go through the test. They stopped their enzyme therapy for 30 days prior to the gene transfer procedure. That enabled us to get blood samples to determine their baseline activity of the alpha-gal. They then underwent donation of stem cells, which was done through a process called leukapheresis. It's like plasmapheresis, where we collect blood cells from the patient and return the cells to them that we don't need. That takes a few hours. The blood sample then was sent to the lab, where we were able to select out the blood stem cells based upon certain antigens or markers that these cells have on their surface. The cells then underwent transduction with the lentiviral vector, which contained the alpha-gal DNA, and they were subsequently frozen. While we conducted a bunch of other quality assurance testing, we had to make sure that there was no infection associated with this construct, that is the virus plus the new DNA, that there weren't any other viruses contaminating this whole process. And the regulatory authority, in this case Health Canada, had to be satisfied with our results. Once we had that, then the product, which was frozen, was simply unthawed and infused through a peripheral intravenous line. And that took about half an hour, 40 minutes. The patients had to be monitored, and they didn't start their enzyme therapy till 30 days after this. So again, it allowed us lots of time to see when they started to make more enzyme and what those levels went up to. And of course, they had to be monitored for any side effects and other safety aspects. And we're currently following patients up to 5 years, but what I'm going to show you is published data out to 1,000 days follow-up. Next slide, please. So the inclusion criteria are here. We had to have males age 18 to 50. They had to have a confirmed diagnosis of Fabry disease with low alpha-gal activity. They had to have a classic type 1 phenotype, which starts in childhood and as they progress, these patients, in addition to angiokeratoma and pain in the hands and feet, often develop kidney disease, strokes, or heart disease. But they had to have good functional status, and we didn't want patients who had advanced kidney disease or heart disease or strokes and things, because they had to be able to tolerate this stem cell transplant. And so here's the results of our 5 patients. It was a pilot study, so we've only done 5 patients. We're not enrolling anymore. We have no more money to handle more patients. Mean age was 38 years. They varied in terms of when they were diagnosed. One was diagnosed at birth, but the others were diagnosed at later points. One patient not till he was 37. They all had classic phenotype, and they had all been on enzyme replacement therapy with an average of just over 6.5 years. And they were on various types of enzyme therapy because the amount that was available varied from time to time. Next slide, please. So one thing that is particular to these ex vivo type of gene therapy is that they have to receive a single dose of a chemotherapy drug. And in our case, we used a drug called melphalan, and we used it in a fairly low dose. And so why do we have to give this? Well, when you take stem cells out of the bone marrow, out of the body, then the body repopulates those stem cells, makes new ones to make up for the ones you've removed. And so we had them in the lab for a couple of months while we were isolating them, while we were transducing them, and then doing all these quality assurance testing. So they were basically frozen. So in the meantime, the body made more. So we wanted to get these blood stem cells back into the bone marrow where they're supposed to live, but the bone marrow's full when it came time for us to infuse them. So we had to basically kill off some of the body's stem cells to make room in the bone marrow for these new stem cells that we were giving back to get engrafted where they can live happily ever after, hopefully. So we did that with the melphalan, and it kills off these cells. And the dose we gave, it was a very short-lived effect, but it was long enough for us to get the bone, these stem cells back into the bone marrow. This was an outpatient procedure, and here's our first patient in January 2017 with Dr. Medine and Dr. Khan in Calgary, and he took the bus home after this procedure. And we follow them daily, and if they get sick, they can always be admitted to hospital. But most of this was done on an outpatient basis, which was good because it keeps our costs lower, and it was easier to get approval for this, both from Health Canada and from our local hospitals. Next slide, please. And so all our patients did well, all 5 did well. None of them had any Fabry disease associated clinical events like strokes or heart attacks during this procedure. They all showed increased activity of the AGAL enzyme in both plasma, that is the blood fluid, and in the white blood cells themselves in the blood. And I'll show you those data shortly. They all had evidence that the new gene was present in blood cells and the gene was active. And there was no change in kidney function, in the urine protein and heart wall thickness, or in other biomarkers during the study so far. And I'll show you some data about antibodies to the enzyme. And they were either negative or they decreased over time. This is patient 4 with myself on the left. And this is Dr. Stephen Kubin, who is our hematologist who supervised the stem cell transplant. And this is Ryan DeVoe, who's going to be talking to you in a few moments. Next slide, please. So on this slide, we're showing enzyme activity. It's 0 down here in the bottom and going up on the y-axis, increasing amounts. And between the dotted lines are the normal levels in blood plasma. And each line represents the data from one patient, 1 through 5. And you can see there's the curves are not all so smooth. They bounce around a bit, but generally they all had low enzyme activity. Remember, they were off enzyme therapy for 30 days. They had low enzyme activity, and the increase, there was a dramatic increase starting around day 12 and persisting after that. And here we are out to 1,000 days. So that's close to 3 years with the first patient. And you'll see that the enzyme level activity has gradually come down. It was above normal for many of them, then it was normal, and has now fallen below normal, slightly in some, but in others it's been a larger fall. But it shows persisting elevation above the baseline, which means that these stem cells are still there, still being active, and still making enzyme. Next slide, please. And we look in the white cells, which are descendants of the stem cells, and we can see there's even more dramatic results where these very high levels indeed in the first several months, and again, a gradual decrease into the normal range in between the dotted lines that you'll see. And the first patient dipped a little bit below that, and then he's back up, and then just slightly under. So just around the lower limit of normal. Next slide, please. And this shows the antibody levels for all 5 patients, again, color-coded. There were a couple of patients who didn't develop any antibodies. There were 2 patients who had pre-existing anti-enzyme antibodies from their previous enzyme replacement therapy. And in both cases, those levels either became negative, 0, or became very low. So this is an example up here of what a high enzyme level would be like. And ours were nowhere close to that. So there's still a couple of patients with very low persisting enzyme levels, but we don't think they're doing any harm in these patients. Next slide, please. In terms of safety, there were no adverse events directly related to the lentivirus construct with the GLA gene. We did see transient low blood counts in all 5 patients due to the melphalan chemotherapy they received, plus hair loss. These are all transient and temporary. There were 3 serious adverse events. There was 1 patient who fainted with insertion of an intravenous line. Another patient had some bruising in the thigh due to low platelet counts. And another patient had fever during the period of low white blood count, but subsequently resolved with antibiotics. Next, please. So in conclusion, this is the first Fabry gene therapy study in the world. It's a phase 1 study which shows that it's feasible and safe in patients. All patients had a sustained increase in their enzyme activity in plasma and peripheral blood cells. But these are preliminary data because the study won't be finished for a total of 5 years. We did see some anti-drug antibodies, but they're very low level in just a couple of patients. But we still need to answer the question as to how much of the alpha-gal enzyme is being delivered to other tissues, such as the kidney and the heart. And we need to know how much enzyme is enough, because we're seeing that those levels in the plasma are not in the normal range. So we'd like to see them ideally in the normal range, but it's highly possible that we don't need to have the enzyme activity actually back to 100% of normal. Patients may be fine with 50 or 60%. We don't really know what the right number is at this point. So there is an ongoing study now using the same technology. This has been taken over by a company called AvroBio, and they're running this study AVR-RD01, which is basically the same protocol. It does involve a different drug. They chose busulfan instead of melphalan. It's a stronger drug in terms of having a greater effect on the marrow, and I hope that they will get larger numbers of stem cells actually in grafting. And they are running this protocol. It's proved in Australia and the United States, and they've enrolled 5 patients with that. And as part of this study, they are looking at the effects of this on target organs. In this case, the kidney. They're doing kidney biopsy at the start and then at 1 year. And they've presented some preliminary evidence or reported some preliminary evidence at a press conference, saying that they had in the kidney biopsy of a couple of these patients, they'd shown that they had removed the GB3 deposits from some of the cells, but they haven't actually presented that data yet at a scientific meeting. And it is possible with this type of ex vivo gene therapy that it could be repeated for patients, and every 5 or 10 years, we don't really know. So you can find more information about this particular trial if you look on the clinicaltrials.gov under gene therapy for Fabry disease, or if you just simply Google AvroBio gene therapy Fabry, you'll come up very quickly to the AvroBio site. I think it's easier just to Google it than for me to give you a long string to click on. And you'll find it there. And AvroBio is developing gene therapy for a number of diseases using this same platform. So I'll stop there, and I thank you for your attention. And I now like to introduce Ryan DeVoe, who I showed you in one of the slides. I've known him for about 15 years. He's a really great person. He is a local IT guy in Halifax for one of our companies. And I've had the pleasure of working with him for the last number of years, first with enzyme therapy and some other studies, and more recently in our gene therapy trial. So Ryan, take it away.
Thank you very much, Dr. West. What I'll do, you've heard all about the clinical side of things. I'm going to go through my experience through the process. So as you heard, I'm Ryan DeVoe, and I've had Fabry since I was very, very young. My parents got me, my brother and I diagnosed around the time I was 4 years old, as you saw in one of the other slides. So I had all the classic signs growing up. I had lots of pain in my hands and feet. I had GI problems, so anything from slightly upset stomach to running back and forth to the washroom for hours on end. I didn't do a lot of sports in school. I had lots of hobbies that were indoors. I developed my career in computers from starting out as hobbies and games as a kid, and then it just kind of went from there. So again, I'm sure a lot of you can relate to a similar situation. I met my wife 15 years ago, and that's when I first started enzyme replacement therapy. I showed the first signs of damage due to Fabry disease then. I had, my heart was slightly enlarged and my kidney function was slightly down. And that was the first of any of my I go every 6 months to get tests done. And that was the first time I ever showed any damage. So we immediately went on the enzyme replacement therapy, and leading up to gene therapy, I definitely noticed a change for the better in my symptoms that I was having. Most of them were gone, but I still would have the odd issues with my stomach. I couldn't really work out or do sports without really having to take it easy for a while after and cool off the best I could. Throwing water on my face, cold water on my hands, that kind of thing. And of course, still some pain. Leading up to gene therapy, I had one son then, and we were looking at having a second. So we had to delay looking at the gene therapy until after that, because where there were risks that I may not be able to have a child after, even though I did have the option of sperm banking just in case, we wanted to get that part of our life out of the way first. So after my second child, he was about 1.5 years old, I did go in and do the gene therapy study. There's lots of things to consider as a patient. There's all the side effects potentially that could happen. There's the time commitment of your appointments. There's the time commitment of doing the actual process, and potentially things that can go wrong. The worst of all the things that could go wrong, of course, is you're looking at a percentage chance that you might die in the process. So all these things have to be considered. I felt that I was healthy enough to do this, and so did the doctors. My wife and I had extensive conversations. I asked hundreds of questions to poor Dr. West leading up to it. He was very, very helpful, answered every question I had, and we started the process. So this was 2018, so I'll just do a kind of a quick as I go through. So in September, we met, we went over everything for the actual, I am saying, yes, I want to do the trial, let's start the process. And then I had to go through all my standard tests to see if I qualify for the trial. So all the things you would normally do, the GFR, the MRIs, blood tests, and I had to get all those done. Once they were sure I qualified, we had to go, and then we had to get me set up to do the trial. So I had to get what's called a Hickman line put in, and everyone's experience may be different. Mine was something that goes in your chest and then up in a vein and then down to your heart, basically. So you have basically like an IV hanging out of your chest for me, it was about a month. So when I had to go in to get that done, I was awake for it. They just gave me some drugs to relax me. Years ago, I had a horrible fear of needles. So as you can tell, this was a big stretch for me. And it was one of the things that I was worried most about in the process. And it went really well. I had no issues with it. The doctors were great. The drugs they gave me to relax me worked great. So there was no issues with that. It was patched up. And they basically would check me anywhere from every couple of days to, and then once a week, to make sure that all the dressing was okay. And there was no infections or anything from that. After that, we had to prepare to get my cells out. So they gave me a drug to speed up the stem cell production. So as a patient, I felt some pain in my bones, almost like when you get a cold, but quite a bit more so. It wasn't too bad. I just took a bit of Tylenol and I was okay, but I surpassed I've got a cold and my bones hurt after about 2 hours. So I sat down at the computer to do some work. I can't remember what I was working on. And by the time I got up, I really noticed it. So after they did that for about 5 days, I got an injection every evening or every day, depending on the day, to continue that process. And then I went in for the apheresis. So that's the part that Dr. West talked about, where they take the stem cells out. So I was basically sitting on a hospital bed for about 4 to 5 hours, I believe. And you just sit there and there's a big machine next to you and you watch your blood leave your body, spin around in the machine and then go back in. So it was quite the process, but I had no issues with that. They monitored me closely. I just sat there and I did things to keep busy for that long. And that was, I left after that, and they sent my stem cells off to the lab to get modified. The other thing that so there's two things that I was really worried about during this trial, in the sense of I didn't have any experience with it. So it was just kind of looming over me. One was getting the Hickman line in my chest that I talked about. The second was getting bone marrow taken. I had to have 2 bone marrow biopsies, which basically I had to lay on a bed, and they numbed the site and used a needle to go into my bone and pull out some bone marrow. And I would say I never went above a 6 out of 10 on the pain scale. So in my eyes, that was the best it could have gone. I was totally fine with it. It's better than I expected, because the first thing when you talk to anyone about bone marrow is everyone has their own horror story about what could happen or how bad it was. But I tried to kind of ignore all that and stop everyone. And it wasn't that bad at all when I had it done. So once that was done, it was just, I went home and just waited to go and do the Malphilan. So that was the chemotherapy drug that Dr. West talked about. So I had to drink a whole lot of water beforehand. You could tell in the picture that Dr. West had shown that I was quite bloated. Like my face was much larger. Like I definitely was, I can't remember how much they put in, but if it was through IV, they put in saline. So I was definitely bloated at that point, but I felt fine. Everything was going good. They put in the Malphilan. So I just sat in the hospital and just through IV, and everything went fine. It was just like I was getting my enzyme replacement therapy, and we just went home. We went home, and I had to go through some safety protocols for example, when you take Malphilan, you can't use the same washroom as anyone else in the house. You can't kiss anybody or touch anyone, or basically no bodily fluid transfers. So because the Malphilan could impact them or make them sick. So it was just separating myself from touching other people, basically, which really, it wasn't that hard to do. That didn't bother me at all. The transplant the next day, we went into the hospital. Again, lots of checks and things on me to make sure everything was going okay. And when we were actually doing the transplant, they took the stem cells out of the freezing unit to thaw them. And it was like something out of a science fiction show. And it was just like the moisture coming off of it when it came out. It really did look like something out of a Star Trek or Star Wars. So really cool to see. I was interested in the process, but anyway, so once they took that out, they thawed it out and they put it in the IV bags. And they told me that it would feel like there was someone sitting on my chest because the preservative for the stem cells is processed through the lungs. So it definitely did feel like there was someone sitting on my chest. I had to concentrate on breathing deep. I knew everything would be okay. I was just kind of telling myself that in my head. And again, it definitely was, I'd say, a little bit dramatic, but it went good as long as I just kept calm. And that was done in about 12 minutes, I believe it was. And then that was it. We basically left the hospital. My wife and I went home, and they had warned me that my breath would smell like cabbage because the stuff that is processed through my lungs from the stem cell preservation would smell like cabbage. We weren't prepared for how much I smelled like cabbage. We got in the car and my wife almost threw up. So we had to open the windows the whole way home in November, which in Canada is very cold here then. So it was, it's a funny story, but it didn't cause me any discomfort. We literally just went home. I went about my normal life for, I'd say close to a week. And then my numbers started dropping for my blood count. So where I have kids that were in school, of course, they're always bringing home runny nose or sniffles or something. So Dr. West and I decided for myself, it would be safer to go in the hospital for the time while my blood counts were low. So I was admitted to the hospital and I was there about 6 days. And I just got really, really tired. There was no pain, no discomfort, no issues. I was just really tired. I just went there and relaxed, recovered, and just let everyone do whatever they needed to do to make sure that I was okay and that I was safe. I did have one small incident where I almost passed out, or I did pass out. Basically, I was really, really hot. I wasn't drinking enough water, and I went out to get some water, ironically, or a snack or something, and they wanted to take my blood pressure, and I just fainted there. So I did hit the ground, but not too hard. They ran some tests and everything was fine. It wasn't an infection or anything like that. So I have had that happen once before, just when I overheated when I was outside doing activity. So I personally think it was just related to that, but it wasn't a medical concern after that. It was all ruled out, everything was okay. That was the only issue I had during the process. After that was done and I went home, it was within about a month. And I started to notice that I had more energy. It was coming up to Christmas. I noticed my energy was coming back. And then there was a point where I felt better than I had felt before I had the gene therapy, energy-wise. I never considered myself to be someone who suffered from low energy or had energy problems. I never thought that. So for the first day, I just kind of thought, okay, well, it's just an off day. Maybe it's in my head, because you're second guessing everything. You expect to feel good afterwards, or at least I did. I went in thinking that I would feel better after this, but it starts playing on your head a bit. So day 2 and 3, I started to say, I really do feel better. I have more energy than when I went into all this. And sure enough, that was around the time that my enzyme levels, after talking with Dr. West, they were at or above normal. And since then, my energy has stayed the same. I feel better day to day. Everyone's experience may vary if they do go ahead and do this, of course. Any of the outcomes I'm describing now are obviously my personal experience. So Christmas went good, basically. That's when that happened. I had more energy. I never had any of my pain come back. My GI problems, over the course of months, they never got worse, and they just got better from there on. I went in November 21st for the transplant, and I went back to work January the 1st, and I was back full time by the end of January. So I think I personally, I handled it really well. Like, I was surprised at how quickly I kind of bounced back from everything. I did lose hair. My hair's thin anyway, but I did lose most of it in the process, and I grew back within a couple of months. It started really growing back. That was the only noticeable effect it had on me, other than making me really, really tired. Now I'm working out 5 days a week in the morning. We get up early. I'm doing intense work. I still don't sweat, so I need to have, we have an air conditioner that blows cold air on me the whole time because I can't sweat. But I recover faster, I feel better. I'm noticing actual changes in my body from being able to do these activities. I was never really overweight, but I was definitely not in shape. So I'm definitely noticing these changes now after being able to do this. And I just realized about 4 days ago, I'm not running to throw cold water on myself anymore. And I was, even after gene therapy, I'm not running after workout now to like that I'm feeling completely dead. I have to cool off, or I'm going to, I'm going to have to lay down or something. I'm just recovering faster. I feel better. I've never been able to stick with a workout program this long in my life. Even though I still don't sweat, it doesn't completely destroy my day by doing all this. That is the very quick story of how when I went through gene therapy. So I know I probably missed a few things. I'm happy to have any questions sent to me. I started my life very closed with Fabry disease. I didn't tell anyone outside my family until I was 23. And I really regret that decision. So now I try to talk about it as much as I can. We are rare, and unless we talk about it, nobody else will. Not enough people will. So if there are questions I can't get to today, please send them to the organizers and they can get them to me through email, and I'm happy to help answer anything I can. So for now, I'll finish off and I'll turn it over to Atul, who is going to, I believe, go through the questions and answers. Thank you very much for inviting me.
Okay. Well, thank you very much, Ryan. That was really fantastic account, and it's really great to see you looking so well. And it's great to hear from Michael, Darylyn, and Sophie. I think we've got some questions. Do you want to pose them? Have we got time?
Yeah, no, absolutely. So we've had a few questions that have come in. So my first set of questions is to go to Michael. And one that came in during the webinars was, should babies born to Fabry patients have a sample of stem cells taken and stored as a matter of course?
I'm sure that's technically possible, but unnecessary. We can much more easily access these in adults, and would be, I think, less traumatic to be taking them from an adult than from a newborn. So we can, it's not necessary to take them from young children.
Okay, thank you. And another question for you, Michael. We had another question during the webinar, and we had one pre that, and Darylyn has responded to one of them. But women can also have severe organ issues due to Fabry disease. And so do you expect women to be included on these trials? Because obviously a lot of them at the moment are looking for classical males. So would it be the plan to open it up to females as well, and also children? We've had some questions through as to whether gene therapy will be available for children.
Well, first of all, I'll answer the question about women. So yes, the AVROBio, I believe, has an amendment to their study, which means they will be able to enroll patients men who have previously been on enzyme therapy, because they were just looking for men who had never had chaperone or enzyme therapy. So they're opening it up to men regardless of their prior therapy, and they're also going to, I believe, include some females. And the issue around including women is that, of course, some women with Fabry disease really don't have any symptoms whatsoever or signs of the disease. They are carriers and can pass it along to their children, whereas others, as we all know, are severely affected. I have one woman who's had a heart transplant, for example. We have other women with kidney transplants. So yes, it can be quite severe in some, but trying to sort them out is somewhat difficult at times. So, so there may be some other specific criteria these women will have to meet for these trials, and I don't know what those will be as of yet. In terms of including children in these trials, I think that we need to show efficacy, that is, how effective this will be in adults before there will be consideration of putting children into these trials. But I would point out that the trial I mentioned in adrenal leukodystrophy, the other lysosomal disease with a lentiviral gene therapy, most of those subjects in that study are in fact children. So there's no reason a priori why children can't go into this study, but I think we need to show that it's effective in adults first before there would be a separate trial in children. And again, if you're going to be doing this in children, we need to know that this stem cell transplant would be safe for children, and it certainly is done in children with hematologic diseases like leukemia. So it's certainly possible to do it.
Great, thank you very much. So Darylyn, I've got some questions for you, if that's okay. Obviously, at the moment, we're still wrapped up within the COVID pandemic, and we've had a question about whether clinical trials are fully open, and how safe is it currently to attend hospital?
Thanks, Sophie. So it will vary a little from site, hospital to hospital. So at our hospital, we are fully open. There are rigorous checks before people come into the hospital. So you would expect to get a phone call before to make sure that you are well, and then a temperature check when you come up. But we are open, the trials are open. We're recruiting to all of the studies, and the transmission rate of COVID is pretty low generally at the moment, and the hospitals are very safe. So I would encourage you to go to your appointments and also to come up for clinical trials if your clinical team asks you to do so.
Thank you, Darylyn. And another question, whether Darylyn or Brian, you might want to answer this. Given there are multiple delivery methods for gene therapy, what should I consider when deciding which trial would be right for me? Darylyn, let's go to you.
So I think broadly, there's the in vivo versus the ex vivo, and they're quite different in terms of whether you want to have some of the procedures that have just been described, so stem cell harvest and a little bit of chemotherapy, and then the stem cells going back into you, versus having the infusion of the virus. So there's something to discuss there with your clinicians about those two and which one might be right for you. And I think you know, we've got to do a lot of work in both areas to understand in the longer term whether we'll still be using both or just one. But I think they both seem like really good options, but some might be better for one person than another. Within the viruses, I think we don't know enough whether it's possible to recommend or even think about how one particular viral vector might suit somebody better than another one. So again, I'd recommend that you talk with your clinicians about what the latest results are, and then you have a look at what's going on in the specific trials at the time that you might want to participate. So where are they recruiting? Are they recruiting the sort of patients that we've been talking about, classical males, or has it been extended? And then also, I think importantly, where are the studies in terms of the dosing? So what sort of doses are people getting in the current cohorts, and what are the results from those if your investigator knows about it?
That's great. Thank you. Brian, do you have anything to add to what Darylyn said?
Not very much, really. I mean, some people have been exposed to adeno-associated virus and may not be able to take certain serotypes, and that's something else that has to be taken into account, actually, in the initial assessment.
Yeah, absolutely. Thank you very much. So we just got time for 2 more quick questions. Another one for you, Michael, if that's okay. If gene therapy is not effective, would I be able to continue receiving enzyme replacement therapy?
Yeah, the short answer is yes, or chaperone whatever someone might have been taking. And that's certainly the case in Canada. I would note that 3 of our 5 patients have stopped their enzyme therapy after the procedure. 2 are continuing on that. And we stopped in those patients because we felt under the circumstances that it was reasonable to do that, but it's always an option to restart enzyme therapy at any time should the patient need it.
Thank you. And our last question is to you, Ryan. What should I consider when choosing whether to enroll on a gene therapy trial? I think it'll be really helpful to sort of hear from a patient who's made that decision. Obviously, you've spoken about your individual choice, but what are the things that you would be advising patients to consider?
Depending on the conditioning drugs, of course, as I mentioned, children, where you are in your life when it comes to children. Will it be safe to try to have children after? So definitely talk to your doctor about that. For me, one of the things I was considering as well was time. I was doing 1.5 hour, 2-hour infusions and every 2 weeks, and now I haven't done them in 1.5 years. So I've gained back all that time. Safety, I had, of course, questions around my safety, which I kind of went over some of that already, but you need to advocate for yourself, ask questions to find out, you know, is there anything specific to you that you should be concerned about? Don't go into this with any unanswered questions. Dr. West was wonderful with me. I bombarded them with questions. This is your life, and you need to do any questions you have, ask them. You need to worry about just some logistics, getting to and from your appointments. I couldn't drive for a bit after. I felt good, but safety-wise, I was told that I'm not allowed to drive. So I had to get other people to drive me to and from appointments. What to kind of worry, what to ask questions about kind of varies for different people, but that's kind of the general idea. If you have anything specific to yourself that you're not sure, just ask, basically. I know that's a very broad answer, but once you read and go over the material with your doctor, most of the questions for me kind of jumped out at me. It was just risks, safety, logistics. Children was big for me. We definitely wanted to finish that part of our lives first.
No, that's really helpful. Thank you so much, Ryan.
No problem.
Well, that's all the time we have for questions. If we haven't answered any, we can get that across to you. And as all of our speakers have said, if there's anything specific that you want to ask them, then do direct it through the MPS Society and we can forward it and get the responses for you. So that's all that's left for me to say is thank you all for joining this webinar, and to thank all of our speakers for a very insightful, informative presentations, and hopefully it's given you all a little bit more information about the current gene therapy trials available, and to also give you an experience from Ryan, a patient himself. As mentioned, this webinar is being recorded and will be available on our website shortly. And we would really value any feedback that you have in respect of future webinars that you would like us to host for you. So from me and from all of the team here, we would just like to say thank you and goodbye.
Sophie, night.
Good night.
Thank you, everyone.
Thank you very much. Thank you.
Thank you. Bye.